Hello, we were going through the survey of fungal module. So today's agenda will be to see PUCCINIA, that is the fungi which will be causing stem rust or leaf rust in wheat plants. So we officially start Basidiomycota today. Before we move on to today's agenda, let us go through the usual exercise. This is the survey of fungal module that we will be seeing. First of all, we have to see Zygomycota that we have already completed with the Rhizopus and Mucor life cycle. Then Ascomycota, within that we have seen Saccharomyces, Aspergillus, Penicillium, Neurospora, Claviceps and Precisa with detailed description and interesting videos. Then we are officially starting with Basidiomycota, PUCCINIA. Then we'll be seeing by tomorrow or maybe by today itself Agaricus and Ustilago. Then after that we'll be moving to fungi-like organisms within which phylum Oomycota. We have to see life cycle of Phytophthora and Albugo. Then we'll be seeing some general topics like lichens, mycorrhiza, fungal toxins, etc. So have you observed these kind of images in Google or even in the real-time fields, etc.? These kinds of brown colored structures which are seen on leaves, these are called Eurydiosaurii or pustules. Okay, this orange, red or brownish colored like saurii that you will be seeing, this is completely filled with the spores of the fungi. That spore is called Eurydiosaurii. We will be seeing. Okay, so you can see a different colored spores, black colored spores. So what is this thing? Okay, then you can see this bicelled, okay, two-celled structure. What is this thing? So that means the life cycle of PUCCINIA is a little bit complex, but I'll make it very simple. So this is the thing that is the agenda for today. Life cycle of wheat stem rust, that is PUCCINIA graminis triticae. Actually, PUCCINIA is the genus that we'll be studying. PUCCINIA graminis is the species which will be causing this kind of issues for the wheat plant and all. And specifically, if you want to say, the variant of PUCCINIA graminis, that is a subspecies, PUCCINIA graminis triticae, will be causing the wheat stem rust. So we'll be seeing that thing. You can see that there are around 5 types of spores which are in the life cycle of this particular PUCCINIA graminis triticae. So you can say that it is macrocyclic or polymorphic. So it's why it's macrocyclic, because its life cycle is not simple. It is having a very lengthy cycle, right? So macrocyclic. Then why it is called polymorphic? Because it is having 5 types of spores. Okay, Eurydiosaurii, teleospores, basidiospores, pycniospores and asiospores. Okay, starting with Eurydiosaurii, it will be giving rise to teleospores by the end of the life cycle of wheat plant. Then this is during the harvest season, during the summer season, where wheat will be actively growing. Then when the wheat plant will be completing its life cycle, when it is going to die, okay, when it's going to be the stubble, it will know that actually it cannot survive. Now the wheat plant its life cycle is already over, so it will be growing on the dead wheat plant in the form of teleospores. So it will be overwintering. That means it will be coming across the winter season through the spores called teleospores. It is black in color. This was orange in color or brownish red in color. Then it was black in color. Then when the winter season, the extreme harsh winter season is over, these teleospores will be germinating into basidium, and basidium will be carrying basidiospores. Okay, so then after the spring season is over, then it will be giving rise to this particular basidiospores will be traveling to another plant, an alternate plant called Barberries species. Okay, Barberries, this is a different like plant. This is not wheat. Okay, it's a different plant which will be growing in the hilly regions. So it will be this particular basidiospores will be reaching this particular Barberries plant, and on the upper side of the leaf it will be forming pycniospores, and in the lower side of the leaf it will be forming asiospores. This asiospores is something like orange in color. Okay, then asiospores will be during the summer season will be moving to this particular wheat plant, that is then that will be forming Eurydiosaurii. Okay, acidiospores will be forming a germ tube during germination, that will be giving rise to Eurydiosaurii or Eurydiosaurii. Okay, so this is the asexual cycle. Okay, here you will not be seeing any fusion of the mycelium. But during the formation of asiospores, you will be seeing that the two mycelium will be fusing together and will be giving rise to asiospores. So this is in a nutshell. We'll be seeing everything in detail. So you can see that initially the life cycle begins with uridinospores or urideospores. Okay, you can see when you see that this kind of brownish colored pustules. Okay, this raised structure that on the surface of the leaf you will be seeing. If I am taking one particular pustule, you can see that it is a sori. Sori means what? It's an aggregation of mycelium which will be having this kind of spores. Okay, so you can see that it was a vegetative mycelium, and from the vegetative mycelium some of the branches got erect. This is the erect hyphae. This is carrying a structure called orange colored uridinospores. So if you observe the structure, you can see that it is unicellular, only one cell is there. Okay, then you can see that it is dikaryotic. There are two nuclei. Okay, then finally you can see that it is having a stalk cell, right? So it is having two cell wall as well, indine and exine. Two cell wall is also like complex, like that means bilayered, right? So this is uridinospores. Then after the during the harvest season, or when the wheat is actually growing, like actively growing, during this time you can see this particular uridinospores. Then by the time of wheat plants growth cycle complete, okay, after harvesting we have taken away all the grains and all, then the remaining stubble will be left out. Okay, in that stubble it will be growing this particular teleospores. The uridinospores cannot survive the harsh winter condition. You can see the mercury is lower. Okay, that means winter condition is prevailing. Okay, at that time uridinospores cannot survive, and it will be like from now on it will be not growing in this erect hypha, will not be growing this particular orange colored uridinospores, but it will be growing black colored teleospores. Okay, these teleospores, this can actually survive in the harsh winter condition in the soil without having any nutrition and all, because it is harsh winter. Okay, and wheat is already dead. So it will be in this black colored erect structures. Okay, you can see that in the place of uridinospores, now it has the uridinospores is not forming in large numbers now, but teleospores are forming in large numbers now. So what is the difference between uridinospores and teleospores? This is why I have inserted images in my videos. You can see that uridinospores was single cell, it is bicelled. Okay, and uridinospores was like orange red in color, but it is actually black in color. This black colored streaks will be formed on the stem, leaves, etc. And it is actually each cell, I told that it is bicell, and each is actually bikaryotic or dikaryotic. You can see actually in some text you can see that it is something kind of fused structure, but it is not fused yet. It is in the dikaryotic stage itself. I'll show you in different images why it is not fused till now, but it is in the mature stage. That's why it is showing this fusion, but in the initial stage it will be showing this dikaryotic stage only. This is the mature form. That's why it is showing as fused. Okay, fine. Okay, in this place of uridinospores, now you got the teleospores. Okay, now these teleospores now come here. Okay, during the time the winter season is getting over, that means you are reaching around the summer season, or spring season. This spring season you can see that this will be forming wait a minute, this will be forming basidiospores. Okay, one second. Yeah, basidiospores. Okay, I told that these particular teleospores were forming on the dead wheat plant in the soil. So in the soil, this during the time of spring season, these particular teleospores, the top cell and bottom cell, both will be forming through a germ tube. It will be forming a small mycelium called basidium. Okay, it will be having 3 to 4 cells in length. Okay, and from each cell, like each compartment of this basidium, there will be the formation of a sterigma. This projection is called sterigma, which we have seen in like Penicillium, agaricus, aspergillus, etc. This sterigma will be forming a projection to outside, and to that projection there will be the passage of nucleus. Before that, something will happen that I will show in the explanation part. Okay, when I am explaining the drawing. Okay, this particular nuclei, I told that it was in the dikaryotic stage. Now it got fused. Now this is the situation. This fused nuclei will be undergoing meiosis, and we know that when one meiosis occurs, there will be creation of 4 nuclei, right? So to each of the compartment that is created here, there will be the passage of this like haploid nuclei towards the basidiospore. Okay, this basidium, it is having a projection called sterigma. Now it is having a circle shaped basidio spore. Okay, towards this basidiospore, 4 haploid nuclei will be moving. So that will be forming the basidiospores. Now the basidiospores will be during the like spring summer season, beginning of summer season, during that time it will be moving to this particular structure like barberry plant. Okay, barberry plant which will be growing in the hills. This is the alternate host or second host of the particular like Puccinia graminis. So on the surface of this barberry plant you can see a flask shaped structure. This is the upcoming basidiospore. It is falling on this particular barberry plant. On the surface of the leaf, on the top surface of the leaf, it will be forming a flask shaped structure called spermagonia. Spermagonia. Okay, it is also called pycnidium or pycnium or pycnidium. And from the bottom of the pycnidium or spermagonia, we can see some erect cells, erect hyphae is growing. This is known as pycnidiophore. Okay, why it is called pycnidiophore? Because it will be releasing pycniospores or pycnidiospores. Okay, this pycnidiospores is also called spermatia, and you can see that there will be some hypha which are called receptive hypha. Okay, we know that this particular basidia was created out of 4 type of haploid, like 4 haploid nuclei. Out of this, 2 are of plus type and 2 are of minus type. So there will be 2 types of flask shaped spermagonia will be created. This is of plus strain and this is of minus strain. So the spermatia from the plus strain will be reaching the receptive hypha of minus strain. Similarly, the spermatia from the minus strain will be reaching the receptive hypha of plus strain. So that will be causing some sexual fusion, right? So it will be a kind of sexual fusion. Then from the pycniospores, this got fertilized. One strain got reached the hyphae further and both got fertilized. Okay. So while this fertilization was happening, something was happening on the lower side of the leaf. This happened on the upper side of the leaf. Towards the lower side of the leaf, you can see that the mycelium from the pycnidium, this is actually mycelium. Okay, this is hyphen structures. The mycelium from the pycnidium was coming towards the bottom side and it was creating an orange colored cup shaped structure called acial cup. Okay, acial cup. This structure is called acia. Okay, so what will happen? So this is made up of a monokaryotic mycelium. Okay, that's only what this particular hyphae was made up of monokaryotic mycelium, right? Because it was made up of basidiospores which are monokaryotic. So what will happen now is there will be after the fertilization of the spermatia and the fertile hypha, there will be dikaryoticization that will be happening. Okay, that will be the formation of dikaryotic mycelium, and each of the nuclei from the dikaryotic mycelium will be reaching even the acial cup, and that will be making even the acia dikaryotic. So this acial cup became dikaryotic because of the fertilization and the nuclei coming towards this particular acial cup as well. So this acia now became dikaryotic, and from this dikaryotic acial mycelium there will be the formation of dikaryotic aciospores. Okay, I'll show you here. This is orange colored structure called acia, and here you can see that dikaryotic from this acial mycelium, it is dikaryotic aciospores is formed. Okay, this aciospores will be still it is in barberry plant. Okay, now it will be released towards like during the summer season it will be released, that will be moving towards the wheat plant, and it will be forming it will be reaching the surface of the wheat stem or leaf, and it will be forming erect hypha, and it will be carrying euridiospores. So from the aciospores it reached the wheat plant, it formed the euridiospores. Okay, so that will happen. This, like as I told, aciospores will be germinating to produce euridiospores, and that will be the visual that will be seeing on the as the stem rust in the wheat plant. So this is the summary of the entire life cycle of this Puccinia graminis. So in the upcoming video I'll show you a little more things with drawings. Okay, now you just see this is a summary. So it was euridiospores. It slowly during the end of growing season it gave formation, gave the formation of teleospores, which is bicelled and black colored. Then teleospores on the soil gave rise to basidiospores. Basidium released basidiospores. It reached the barberry plant. It formed the spermagonia. Then it formed pycnidiospores and then acetospores, like acetospores, which reaches the wheat plant and formed the uridiospores.